Okay, we're going to be continuing our blood lectures for Bio 232, and we're going to be looking at hemostasis today. And if we have time, we'll also get into red blood cell formation, red blood cell typing, blood typing. We'll see how we do. Let's switch over to the lecture notes here. So this is where we finished last. Bottom of page 10, hemostasis. So hemostasis literally means blood stopping or a steady state of blood. So this information starts on page 657, and goes all the way to 661. Now, the book has a lot more details in it than we are going to cover, but we're going to hit some of the big parts, okay? So be aware of that. Now, there are three main parts of having to do with hemostasis. There's vascular spasm, there's platelet plug formation, and then there's the actual coagulation or clotting of the blood. And these all happen when blood vessels are damaged or ruptured. Normally, our blood vessels are a closed system, and if we have damage to one of the walls of the vessels, blood will leave, and we don't want that to happen because our blood volume could go down and we could become very ill very quickly. And so we want to stop and close up that hole or that damage, that rip, that tear. And we do that by this process. So the very first thing, so let's just take an example. Let's say you're at home, you're doing a lot of cooking these days, and you accidentally cut into your finger a little bit with a knife. Nothing super deep, but down far enough into the dermal layers where you have hit capillaries and you are bleeding. So now what you would normally do and what you should do is go and immediately rinse your finger, wrap your finger up, hold and apply pressure until blood stops. While you're doing that, other things are happening. If you could sit there and watch your finger and what would happen, some of these stages would make maybe a little sense. I don't recommend this though. So the first one is vascular spasm. And vascular spasm is triggered by smooth muscle damage in the walls of the vessels. Okay, so that's the first thing. When we cut into a vessel, we're going to cut through the smooth muscle layer. We're going to talk about vessel anatomy in probably the next lecture or two. And we're also going to cut through the endothelium, and the endothelium is that epithelial lining of the vessel. And so when we cut through those, we're going to release chemicals, and those chemicals are going to alert the body that we've damaged that area and we need to repair it. Platelets, the presence of platelets will trigger vascular spasm, and the pain receptors will trigger vascular spasm. So what does vascular spasm do? Well, all that vascular spasm means, I'm going to flip back over to me here, is that you've got a vessel, there's the lumen where the blood would go through, and now let's say we tear, here I'll just use one hand. So we have a small vessel and we have damage, and so we rip the wall like this, and blood is now pouring out into tissues, or in your case out onto your skin, and we need to get this so it's closed up. So the very first thing we do is around the actual in the wall is smooth muscle, and so what we will do is we will tell the smooth muscle to constrict, and as it constricts, it'll bring the edges closer and closer together. Now, in an ideal world, they will completely get next to each other. They might not be able to do that, but they will try. So vascular spasm is causing these vessels to decrease in diameter. The lumen here will get smaller, and the walls will come close together. That way we can go ahead and we can actually now try to connect those walls together. So the first thing this will do is it'll reduce blood loss for the first 20 to 30 minutes, and it allows time for other hemostatic activities to kick in. So if you were to have cut your finger and you were just to set your finger there on the table and watch, what would probably happen is a lot of blood would come out initially, and then over the course of a minute or so, the blood flow would slow down greatly, and this is due to vascular spasm. Blood still may be coming out, but a lot less than the original. Okay, so the greater the damage to the vessel, interestingly, the greater the vasoconstriction. What we're going to notice in hemostasis is there's a lot of positive feedback stuff happening. We don't want to just have a small response and go back to normal. We want to have an exaggerated response. And remember, in positive feedback, we kind of keep stimuli makes us go farther and farther away from normal. And we want to do that here because we want to stop blood from flowing. We don't want to just slow it. So this is an example of that. The more damage that we see, the greater the vasoconstriction usually. And obviously up to a point, can't do, it doesn't work for ever and ever that way, but this will help slow blood loss. Okay, so that first part's pretty easy to understand. The second part is platelet plug formation. And platelets are floating around in your blood all the time. Small amounts of platelets are floating around. And normally they don't interact with things. But if they come in contact with the collagen fibers of the subendothelium, they're damaged blood vessels that are sticking out, the platelets will enlarge. They'll become irregular and spiky, and they will become sticky. Okay? So they will get bigger, they will get spiky, and they will get sticky. All right. They also start to degranulate, meaning that the granules inside will start to release the chemicals. Okay. Now, some of the chemicals we talked about at the end of last lecture are now showing up. Okay. So you need to know the names of these and what they do. Okay. So serotonin gets released, and this intensifies vascular spasm. So it's a chemical that tells the vessels nearby that have damaged walls and have little collagen fibers stuck out to intensify vascular spasm. ADP, spent ATP molecules, attracts more platelets. So this is again giving like a positive feedback loop, and it makes them sticky. Okay. Thromboxane A2 does both those things. Okay. I'll put that in a different color just so it's easy to see them. Okay. Does both. All right, so that's important. So most of these chemicals will release all at once, but if only one is released, then you'll see one response. If thromboxane is released, you see both responses. Okay, and this will start a cycle that will create what we call a platelet plug, because as one platelet, so I'm going to draw over here. And by the way, there is a good picture. Here, before I draw anything, let's go to your notes, not to this one, to up here. These are the 3 steps, of hemostasis. 4 pictures, but 3 steps. This is on page 658, and we're right now in platelet plug formation. And let's zoom in. Maybe I can just show this. And so what'll happen is here's little collagen fibers that are stuck out in the subendothelium, the part below the lining of the vessel, and platelets will start to stick to them, and as they do, they will release chemicals, and that will cause more platelets to stick, because remember, the platelets become spiky and sticky, and so more and more platelets that are floating by will start sticking to this, and it will start to block up this little gap. So remember, the gap was a little bigger at first, and then we vascular spasmed, and now we have a small gap, and now that gap is being filled by platelets. Okay. However, this is good, but it's not perfect. We need to completely close up this area so we can repair this underlying wall and then get things back to normal. So one way I think about this is the platelets are kind of like sandbags. And so if you have like a river that's overflowing and you put sandbags down, the sandbags do a really good job of not allowing water to go through them. But some water does go through the sandbags. It just slows it significantly. So now what we want to do is build like a more, even a better structure that has more, meshwork and more things on it that will hold those sandbags in place and will let even less fluid out. And that's our 3rd step. And that's where we're going to go next. So by doing this, by having these platelets come in, we're getting to cause a platelet plug, and we're getting positive feedback. Now, we don't want to make a plug that's too big, okay? And so we do have a chemical, prostacyclin, that is released, and makes sure that the plug is only limited to the specific area of damage. I'm going to write the word of damage here. By prostacyclin. This is good because if we accidentally started to plug up other areas, we might plug up healthy vessels, and we don't want to do that. So we only limit the plug to the specific area. Okay. Now the third thing that we do is coagulation or blood clotting, and this is a very complicated process. There are many clotting factors that get involved. There's a big cascade, and I was going to show you how complicated it is. If you go down on to, well, here's all the clotting factors listed. You don't need to know all those. And here's the entire clotting pathway. Look how complicated that is. That is on page 659 of your book. You don't need to memorize this. Where we're going to start is right here. We're going to start here and just have you learn this bottom part here, the end of phase 1 and phase 2 and phase 3. Okay, but if you're missing these clotting factors, you can definitely have problems with this whole cascade. For example, people with hemophilia are missing certain clotting factors, and those clotting factors then don't allow them to clot their blood very effectively, and they can have very life-threatening problems. But we're going to kind of skip all those. Okay, so if you flip back to our notes, you'll notice this part here is similar to the bottom of that page, and we're going to type in, we're going to actually put some stuff in here and talk about this a little bit. Okay, so what happens in blood clotting? So there's 3 major events. There is different activations of this. And so what I'm going to do is I'm going to read this, and then we're going to fill it in and add a couple extra things in. Okay, so first thing that happens is we have this prothrombin activator. Now, whenever you see a word pro in front of something, it means that it's a chemical that is not actually active at that moment. Okay, it's a precursor molecule. So prothrombin is not active thrombin. So prothrombin activator is something that is going to activate prothrombin to thrombin. So we're going to make prothrombin activator. Okay, excuse me. Here, let's erase that. We don't need to do the intrinsic extrinsic part. We can skip that. Prothrombin activator. Okay, so prothrombin is converted to thrombin by prothrombin activator. Okay, so where would we put this on our little thing? So first, let's read what's happening here. So it says we're getting some chemicals and some clotting factors from the platelets, and there's calcium. All right, so let's that is needed. Underline calcium and vitamin K. And remember, vitamin K helps with DNA and eventually protein formation. And so we need vitamin K also. So we'll put vitamin K and calcium and other clotting factors. And these things will result in making, I wonder if I can make this a smaller font here. I'm going to pause for once. Okay, well, I'm trying my hardest to make it so I can type in here without making it too messy, but I don't know if I'm going to get there. But I did make the font a little bit smaller. So we have prothrombin activator, all right, is there. And that prothrombin activator, as we said, is going to go and make I'll clean this up in a minute, thrombin into thrombin. Okay. See if I can just move it over a tiny bit here. Okay. Well, that's the best I can do right now. And actually, I'll put these down below and we'll draw arrows because I know they won't fit in. The boxes are so small. So that was this first line. I'm going to go back over here now. So prothrombin is converted to thrombin by prothrombin activator. So now we have thrombin. So what does thrombin do? Well, thrombin, and we'll put this in a different color just again for contrast. Thrombin catalyzes fibrinogen into fibrin strands. Okay, so what is that? Well, you might remember way back when we talked about connective tissue and blood being a connective tissue. Remember, all connective tissues had a fibrous component. But blood didn't have fibers floating around. But we made a comment that there was these subunits of fibers that were only came out during clotting. So fibrinogen is like a little subunit, and these little subunits are floating around in the blood, and so they're not fibers. But when thrombin gets involved, it takes the fibrinogen, which are the more soluble forms, and turns it into fibrin, and we can get these long chains of fibers forming from the subunits, right? And now we have fibers. And those fibers we wouldn't want floating around in our normal blood, because they could cause clots when we wouldn't want them. But we definitely want them when we are in trouble and we need to make we need to clot our blood. So fibrinogen, which is soluble, meaning floating around in solution, becomes insoluble and now becomes fibrin fibers. Okay, and I'll draw little arrows down just so you know that that should go there and that should go there, or vice versa. Okay, I'm going to zoom back out a little bit. Okay, so those fibrin things will now lay over top of the, I'm going to go back to our picture here. There we go, the hemostasis picture. And the fibers will now lay over like a mesh, and that will allow the platelets to be below and to completely lock them in place and to reduce red blood cells and plasma from getting out of the vessel. Okay, so that's very useful. And that's what this says here. So the fibrin strands, the meshwork, will cause formation of a clot. Okay, now while that clot is there, we can now repair the underlying tissue. So that's what's happening in these next steps. So now we're going to talk about clot retraction and fibrinolysis. Okay, so clot retraction, what we want to do is we want to actually make the clot so it gets tighter, and we bring the walls of the damaged vessels even closer together and allow us to repair them better. So what we do with this clot retraction is the fibrin meshwork that we just made will shrink, which is pretty neat that it can do that. So how do we do this? So in the platelets that are inside have a contractile protein called actomyosin. Kind of an interesting name, right? And that's what's going to cause the contraction. And this action, this contraction, will pull the edges of the damaged vessels closer together, allowing the edges to fuse completely. This will squeeze out the serum, the plasma, and it will allow us now to repair the wall. Okay? And the one other way that we get that to happen is, remember we talked about platelet-derived growth factor in the last lecture? This will stimulate smooth muscle and fibroblasts. Remember, fibroblasts make connective tissue, and those things will rebuild the vessel wall. If you recall, smooth muscle can build itself, it can make its own endomysium and rebuild itself, and fibroblasts can rebuild the connective tissue. So we can rebuild and create a new section of this damaged capillary or vessel. All right, so that's good. And the endothelium is replaced by vascular endothelial growth factor. So the endothelium, the lining outside, can be remade as well due to another growth factor, but I won't highlight that one. Now, once we've created and made everything correct on the inside, we need to do fibrinolysis. And fibrinolysis means exactly what it sounds like. Lysis means breakdown or destruction of the fibrin mesh. So we use an enzyme called plasmin, and it dissolves the clots. This also is present in an inactive form. So its inactive form is called plasminogen, and this tissue plasmin activator, TPA, activates the inactive plasminogen to plasmin. And that plasmin then will start to dissolve the entire clot, and it will have take a couple days. It starts with about 2 days after the whole event, and will continue till the job is done. And then when we're done, we'll have a vessel that looks almost exactly like it did originally, with a repaired wall underneath. Okay, great. Now, so I would review that and know the different 3 steps, and then also understand clot retraction and fibrinolysis. We're using some new names of chemicals and some enzymes and things that we haven't talked about before. So you want to know those and understand that material as well as you can. All right, so real quickly, we're going to talk about a couple disorders of hemostasis. And I've kind of alluded to a couple of these, but I haven't really talked in detail. So if we have, remember how we said that normally we limit clotting only into areas where there's damage. If we have clotting in an unbroken vessel, we call that a thrombus, all right, or thrombosis. And the material itself is called a thrombus, okay, the clot. And the problem is if a clot in a vessel that's healthy is starting to form, it can break off. And if it breaks off, it can cause the occlusion of a vessel somewhere in our body, and then that will cause death to the tissue behind it. And this is ischemia, or death to the tissue beyond the blockage is ischemia, all right, because we're not getting enough blood supply. So when a thrombus is broken off, we call it an embolus. So if you make a clot in a vessel that's healthy, and then it breaks away, we now call that broken away thing an embolus, okay? And they don't have to just be broken off clots. Anything that's floating in the blood that can cause an occlusion, air bubbles, which is why we don't want to get air in our lines when we're giving people things through into the body. If you break a bone and fat droplets get in, fat doesn't mix with the plasma and can cause a clot, or any other debris for that matter. And when an embolus becomes lodged in a vessel and cuts off circulation and getting that ischemia again, we call this an embolism. Okay. So a thrombus can become an embolus, and an embolus can lock into occlude a tissue or a vessel and cause ischemia behind it, and that's called an embolism. All right. So, like, for example, if you travel in an airplane and you're going a long distance, they often, like, let's say across the ocean, they will encourage you to get up and walk around or do exercises in your feet, because what will happen is blood will pool down in your legs for many hours of sitting, and in some individuals you can start to get a thrombus forming. And if a thrombus forms and then it breaks off and becomes an embolus, that could go and lodge somewhere like in your lung or your liver or your brain and cause ischemia and can be quite deadly. So they want you to use compression socks, which will reduce the amount of blood there, or do exercises, or get up and walk around every couple hours. So that's one way to prevent an embolus from occurring. Okay, transfusion, I'm not really going to say. You can just read and review about that. And then I do want to go a little bit about ABO blood groups. There's a little in your lab this week too. I'm going to just mention, this should be review from Intro Biology. So I'm just going to kind of say a few things and highlight a couple different things. So the things that you want to know is there are antigens. And we talked earlier about antigens being foreign invaders. An antigen is anything that's on the surface of a virus or a bacteria or another person's cell that comes into your body. and it looks foreign to you because you don't make that particular antigen. They also, in red blood cells, or when we're talking about ABO red blood cells, are called agglutinogens. Okay, and so red blood cells may have antigens on their surface. And I'm going to go to the next page because I think this picture is really good here. So let's say you're type A blood, this top one here. You are going to have antigens on the outside of your red blood cell. They're actually embedded into the cell membrane. There are these like glycoproteins that stick out. And so if you're type A, you will have the A antigen or A agglutinogen on the outside. If you're type B blood, you will have the B antigens only on the outside of your blood. Okay, if you are type AB blood, that's my blood type, AB, you have both A and B agglutinogens or antigens on the outside of your red blood cell. Now, notice AB is really rare. Okay, about 4% of the population, and the two most common ones are A. This is in the United States, and O. So O, type of O, is the most common. About 46% of people, approximately, have it. And notice there are no antigens on the outside. It's O, meaning like the absence of, like zero, kind of. And we don't make A or B antigens on our own blood. Okay? Now, why does that matter? Well, if you have the blood type A, you have an A antigen, you are not going to make antibodies against your own blood, okay? If you did, you would clot your own blood. So that's what this is saying here. You cannot have agglutinins, which is another name for antibodies, that attack your own agglutinogens. You only have agglutinins for the agglutinogens that you don't synthesize, okay? So let's go back and look at that picture again. So if you are type A, you have A blood, or A antigen, you are going to then have B agglutinogens. Now notice the shape difference between A here, which is a circle, and B, which is a square. They really aren't that shape, but it's just for us to understand. These antibodies that are B antibodies cannot attach to those. See, the shapes are different. However, they can attach to B. So if you, an A blood type person, were somehow to get some B blood inside your body would recognize these B antigens as foreign, and the antibodies that you make would attach to them and cause those to start to clump together, and your body would start to try to destroy them. So that is why we have these. So any foreign blood that gets in us that's not like ours, we will attack. Now here, you see that if you're type B, you make A antibodies and would attack A blood, but not your own. If you are like me, you have A and B on the outside, so you can't have either A or B antibodies. If you had either one, you'd attack your own blood. Now, O has no antibodies on the outside and therefore doesn't want to see A or B blood and has both A and B antibodies or agglutinins. Okay? So does that make some sense? I hope. So one thing that then is important to note here is when the body recognizes that agglutinogen, an antigen on a red blood cell, is foreign, an antibody will attack that foreign red blood cell, causing agglutination, which is clumping, and this is an antigen-antibody response. So, what does this mean? Well, it means that some people are what we call universal donors because they can give blood that no one will attack, no one will have a problem with. And some people are universal recipients, meaning that they can get blood from anyone. So let's look at type O here for a second. There are no antigens on the outside of the blood. So every other blood type, including O itself, can get O blood because nothing will attack it. There's no antigens on the outside to try to destroy with our antibodies. This is why blood banks really like to have a lot of O blood on hand because it can be used for anyone in the population. And that's why it is a universal donor, because all other types can get its blood. I am the universal recipient. I am AB. And as a result, because I don't make any antibodies against anything, I can get A blood because I see the A antigen and think that's fine. I can get B blood because I get the B antigen and say that's fine. I can, of course, get my own AB blood. And I can get O because there's no antibodies to even worry about. So I'm the universal recipient. Okay. Now, one more thing. There's the Rh system as well. Now, in the Rh system, what's going on here is, and it's called that because it was first worked out in rhesus monkeys, and so that's where the Rh comes from. So there's this agglutinogen Rh, or sometimes called D, so you'll see it written as either. And what is happening is if you have this antigen, you are Rh positive. If you don't have this antigen, you are Rh negative. Okay? Now, the agglutinins, the antibodies, are not present at birth. Now, all of these agglutinins, the A's and the B's, the ones that are in the pictures here, you have these even at birth. You don't have to make these because you saw B blood. You have them already at birth. That's a really important point. Most of the time, we don't have antibodies already made up. We have to acquire those antibodies by being exposed to something and then make the antibodies to it. But in the A and B case, we have the opposite antibody. But Rh factor is more like normal antibody responses where we have to see the antigen or agglutinogen before we'll make an antibody. And that's what this is saying here. So it's saying normally human plasma does not have the anti-Rh agglutinins or antibody. Rh negative blood must first be exposed to Rh positive blood in order to start making the antibodies. Once the antibodies are there, then we will agglutinate or clump together the blood. So where this becomes the biggest issue is with moms who are Rh negative, okay? Moms are Rh negative, and the baby is Rh positive, okay? Now, that means that the father must have been Rh positive and genetically gave the baby the Rh positive antigen in its genetic makeup. So what can happen is if baby's blood is mixed with mom's blood, mom will see this Rh positive blood, and mom will make antibodies against it. Now, often in first pregnancies, this occurs when birth happens, and there is definitely blood exchange during birth, especially in the placenta regions, because the capillaries of mom and the baby make up the placenta. And so mom will often make antibodies against the Rh positive factor. So if mom has a future pregnancy, if that second fetus is Rh positive, mom's antibodies may attack the baby's blood in utero and cause major problems. So that is the key problem here. Now notice, if mom was Rh positive, she wouldn't make antibodies against her own blood, and therefore whether the baby was negative or positive, it wouldn't matter. If the baby was negative, there was nothing to... Negative means you don't have the Rh antibody, or antigen, so mom wouldn't make an antibody against it. If mom was Rh positive and the baby was Rh positive, mom also wouldn't do anything because the fetus looks just like mom, and so mom won't attack the fetus's blood because it looks just like hers. So it's only when mom is Rh negative and the fetus is positive might we have this problem. And so what do? Well, now, even at the first pregnancy, if we identify that the baby is Rh positive and mom is negative, or we don't know what the baby's state is. So, for example, if the father is negative and the mother is negative, there's no way the baby can be positive. So, but if we don't know the father's blood status, or we know for sure the baby is Rh positive, what we do now is we give the RhoGAM shot. And the RhoGAM shot depresses this immune response in mom and it prevents mom from making antibodies against the fetus. And so we often do this in the first pregnancy now, and we don't wait to future pregnancies. That has solved this problem where we sometimes get Rh negative moms and Rh positive fetuses. All right, so this ends blood. Hopefully that was a good review, especially over that last part. You should know that from your intro bio. So review those. We may ask a few questions on your exam about ABO, and there's some in your lab this week as well. So practice there. That might help too. All right, so great. We're going to move on to cardiovascular for our next lectures. And so we will see you then. Thanks.